Back to another AP Biology video, and today we're going to be going over tonicity and osmosis, which is in Unit 2 of AP Biology. It is not the only concept in Unit 2 of AP Biology, but it is a very prevalent one, and one that you will see on the Unit 2 exam and on the AP test most likely. So we're going to be going over it today. So first of all, osmosis is the net movement of H2O, or water, across a semi-permeable membrane from an area of a high to low concentration. Now I know that might sound like a lot, but we'll be going more in depth to this definition and figuring out what all this means. So here you can see water with some salt in it, and water is going to have a lower H2O concentration, right, than this one, because the salt is taking up a lot of the mass, which means that there's less water because there's more of other things. In this case, there's more of salt. In this one, there's no salt, which means that there's more water. It's just pure water, which means the concentration of H2O, or water, is higher. And now to define this term, a semi-permeable membrane is one that allows some material to pass through, but not all. So in other words, it's selective, right? So here you can see these aren't any specific element, but these blue molecules are not able to pass through the semi-permeable membrane, and the purple ones are able to pass through it. So water and molecules in general, they're always going to move from an area of high to low concentration. Now this is a concept that's very important for you guys to remember throughout this lesson and throughout AP Biology, because it will be repeated in later units. And so I just kind of made a cloud that says high to low, and this cloud is going to keep coming back in this lesson as well, just so you guys can remember it, because it is a very important concept. And so let's see some examples. So here we have a cell as well as a salt solution, which is just water with some salt in it. So we have a cell that's full of water and the salt solution right here. So what do you think is going to happen to the cell when it's placed inside the solution? Hopefully you guys paused the video and thought about it for a second, but we can see that there's going to be more solute in the solution than in the cell. And remember, things go from high to low. Molecules, and in this case water, goes from high concentration to low concentration. This means that there's less water in the solution than in the cell. And it might be a little bit of a perplexing to think about this initially, but all you have to realize is that there's more salt in this water, and that means that there's less actual water because there's more of other things. And this cell just has pure water in it, which means there's no solute, there's no salt, nothing obstructing the pure water. It's just water. And like I just said right back there, more solute equals less water, which means that water will now flow from high to low concentration. It will flow from more water to less water, and it will exit the cell. And here we can see the cell shriveling up a bit, and we'll get into that a little bit later. But in this definition, the solution is hypertonic to the cell. The cell is losing water because it is placed into the solution. And alternatively, we can go the other way around as well. So if the cell had solute, like we see here, there's salt in the cell, and the solution was pure, the opposite effect would happen. So more salt, in this case, there's more solute or less water in the cell, right? Because there's more solute, more salt, more of something else. So that means there's going to be less water because there's no solute in here. And so water will enter the cell. The cell will get placed into the solution, and then water from the solution will enter the cell because there's less water in the cell. So in this case, the solution is hypotonic to the cell. And an isotonic solution is one where solute and water concentrations are equal to that of the cell. So no net movement of water occurs. So imagine you put a cell into a beaker of water, and there's an equal amount of salt in both the cell and the water. So nothing will happen. And so here's the actual effects on the cell in different solutions. So in a hypertonic solution, the cell will lose water, and this will cause it to shrivel up. And this is going to make it lose mass because it's going to get smaller, it's going to kind of get skinnier and shrivel up. So in a hypotonic solution, cells will gain water and swell up. They will kind of gain water, and then water will start pushing on the cell membranes, and the cell will kind of swell up, get bigger, and it will gain mass, and it will become more densely packed with water as well. And obviously, if you put a cell into an isotonic solution and there is no net movement of water happening, nothing will happen to the cell. And so overall, osmosis and tonicity are two very, very key factors in maintaining cell balance, function, and structure. And these are two very, very important things to know in AP Biology and in future science courses that you will take. Thank you guys for watching, and I hope you stay tuned for the next one.